For my PhD, I am working on camera trap data, so I attended the African Field School for Camera Trap Studies, hosted by the Wild Ecology Lab in Kruger National Park in South Africa. It is my first time in Africa, but I was astounded by the cultural diversity in terms of their 11 official languages, most commonly the isiZulu and the isiXhosa, as well as the modern hunter-gatherers, the San and the Khoi. And a bit relevant to my presentation is a country that embraces sexual diversity, being the 5th country to legalize same-sex marriage. So this journey in Kruger National Park informs this preliminary framework I'm going to present on surveying the sexual diversity in wildlife for neuroscience studies. As a brief introduction to sexual diversity, the term sex have often been used to describe the genotypic and the phenotypic traits of organisms in relation to reproduction. But this trait is not necessarily coupled, as this binary and bimodal view elides much of this complexity. Instead, converging evidence suggests a more multivariate view on sex, and my PhD facilitates the understanding of this through constructing stem cell-derived models for understanding human sexual diversity, looking into a rodent species that exhibits sexual behavior independent of sex steroid hormones, as well as investigating sexual behavior in a collective context in these African cichlids communities that are relevant for lacking. So these are just some examples of the videos that I use for these studies. A critical limitation to this approach is that these studies are done in the lab and in captivity, which would affect some of the sexual behavior that are exhibited. Therefore, there is also this need to study sexual diversity in more naturalistic settings in the wild. And these are some examples here. This one is a potential example of prostitution in the animal kingdom, where these chimpanzees perform fellatio on another chimpanzee that were associated with subsequent meat sharing between the couples. In these hummingbirds, this is a white-necked Jacobin, where some of the females develop ornamentation plumages that are like those that are commonly observed in males. This is an example of necrophilia in Himalayan toads. As you can see, multiple males are clasping onto the dead female. And this one is an exhibition of the heterospecific courtship between four sympatric species of jumping spiders. Sexual diversity in the wilds are commonly picked up from camera trap images, and some examples are shown here with the Chinese pangolin, the bush bronzewing, the super lyrebirds, the crested porcupine. And the traditional methods to inspect camera trap datasets are through manual inspection, but as we know, there are many limitations to this approach, especially because most of the images does not contain species or behavior that specific researchers may be interested in. Therefore, part of my framework is to develop automated methods to be able to automatically screen out behaviors of interest. At the same time, that it is important for neuroscientists to understand how camera trap studies are done, because although ecologists provide very important information on which type of species may be useful for studying sexual diversity, there may be some diversity that may be relevant to the brain activity that ecologists may miss out, as well as different goals that neuroscientists have that differs to those of ecologists. Although there are some scholars that postulate that the understanding of the neuroscience in wildlife has important relevance to conservation. Therefore, this definitely needs to be an interdisciplinary approach. One of the potentials to automatically screen camera trap images for the behavior that are present is that we can first pass the images through megadetector, which is used for animal detection, and then pass the results into deep learning post-estimation toolboxes, such as deep lab cuts, which has been used for multi-animal purposes as well as the ability to do individual identification through the specific convolutional neural network. And these are some examples of some use of deep lab cuts. On top of that, they also have this model zoo, which are essentially models that can be used as a plug-and-play fashion for many circumstances. For example, for animals with 4 legs, they have this typical body plane that can be compiled all together to form this super animal quadruped model. Then the pose relationships between these different body parts can be used to infer what sort of behavior may be occurring. And then a bunch of different pose relationships can be compiled together to form some sort of behavior classifier that can be used to automatically screen for behavior presence in the camera trap datasets. Last year, I was in Geneva for several months working with Deep Lab developers to improve the underrepresentation in AI. And that is where I met Sarah Beery, who helped create Megadetector, and this puts me on the path towards working with camera traps. So one of my projects there was combining Megadetector and Deep Lab Cut to apply to camera trap images that we were able to develop this Hugging Face platform. At the same time, I also wrote a Python script that can batch process the entire dataset, although that was done through a rudimentary post-relationship filter. So we would need to have more training data for possibly sexual behavior related post-relationships that can feed into packages like for using action segmentation that allows for a more sophisticated filter. And for these purposes, obviously, it is important to consult ecologists on what sort of behavior may be of interest to study, and what sort of species may exhibit sexual diversity of interest. But for the neuroscientists, the camera trap datasets may provide additional information that may be useful for designing neuroscience studies. One of the dataset I am working with is the snapshot Kruger from the Kruger National Park. And just giving some brief backgrounds of the national park is that it was established by the 19th century president Paul Kruger to protect wildlife, although they also had to displace many Songa communities. So Kruger National Park, the dataset is taken over here on number 34, and it contains the big five of Africa: lion, leopards, elephants, buffaloes, rhinos. And accompanying that, South Africa is also known to have 10% of the bird species in the world. And while there are many animal species in South Africa, their national animal is the springbok. So I was able to experience some of these wildlife through the night and day safari game drives during the period I was there. I was fortunate enough to see many of the animals live in this snapshot Kruger camera trap dataset. And what I did with this dataset is that I obtained the mega detected results, then filtered the images with those that have more than two animals that are above 0.9 confidence thresholds. I also had a different filter that allowed me to get images with overlapping megadetector bounding boxes. And from these filters, these are some of the animals I saw in the datasets. Although I did not observe many interactions of interest to me, except for impalas, where you can see some allogrooming and fighting in these images. And the lack of interactions that I observed may be because the dataset was not conducted during a popular rutting season, or it could be that it was placed on a trail, so that's not where a lot of the sexual behavior occur. It could also be because I put the confidence threshold too high, so that I was missing out on a lot of behavior. From this camera trap image, I passed it also through the deep lab cut model zoo, but as you can see, some of the poses are not very accurate, so I'll talk more about that later. But on top of the interactions that we find here, we definitely need more camera trap datasets exhibiting sexual behavior. So I will be specifically looking into literature that captures sexual behavior in camera traps, that enables me to pass the post estimation and then feed all of the portfolio of sexual behavior poses to train behavior models for filtering out camera trap datasets with sexual diversity present. It is also beneficial for me to understand how camera traps are set up. So along with other researchers from 13 countries, we were setting up the camera traps in the field. And we had researchers from places like Congo, Tanzania, Colombia, Madagascar. So we became familiarized with the type, the components, and the features of the camera trap, including the passive infrared, the detection zone, the trigger speed, the recovery time, the field of view, as well as whether the camera trap enables flash, enables color, night mode, correcting for motion blur, and the different burst settings, the sense levels that were used for these camera traps. And we also did some pre-testing of the deployment settings, including the walk test, and considering the different locations for camera trap placement, as well as the height and the direction to the microsite of the species of our interest, as well as how to secure the camera traps. So these are some of the camera trap images we were able to get from this fieldwork. So these camera trap data can be run through Trap Tagger for object detection and species classification that gives me an idea of what are some of the possible species that could be used to study brain activity. And in favor of this, there has been a few studies that examined EEG recordings in wild animals. This one looked at the dusky endocrinus in terms of their sleep architecture and regulation. This one in the Australian magpies as they were solving reversal learning tasks, and this one the elephant seal during their dive. These type of studies are now becoming more feasible as the neuroimaging devices become lower in cost, portable, wireless, cloud-enabled, and smaller. So with the camera trap datasets, domain knowledge, as well as combination with other datasets, these can provide important information on what species could be used to study their brain activity by assessing the feasibility and the design of the neuroscience studies. For instance, considering the scale size of the animal, what sort of behavior that may be present that could disrupt the device, the estimation of the conditions of the animal, as well as possible human-wildlife conflicts and phenology that provides context of the neural recordings obtained. While I have not chosen a specific sea species that I'm going to do comprehensive camera trap data analysis, I've gotten help with the existing data sets on what type of analysis that I'm able to do on that existing data, or other additional preparations I would need to add on top of those data sets. But prior to that, these are some of the existing camera trap data sets that capture sexual behavior that I think would be of good reference to me for my purposes. And this example is the golden tarkin, where they captured their sexual behavior and then measured their activity indexed across these 2L bins, which can give us insight into what period of time would be best to capture sexual behavior. This study was looking at the community in the Borneo rainforest, and they captured the sexual behavior of these small-toothed palm civets. And this graph is showing the number of species in the dataset, which can provide the context of the complexity and the diversity in the environment. Camera traps can also provide occupancy data, and this study discussed about the Samba mating system apropos to how it has been disrupted from hunting and the observations of the decreased amount of mating interactions at low density of the Samba. They also discussed about how salt lick may attract surrounding animals that can facilitate mating interactions. So these graphs are showing the occupancy probability of several different species in the Thailand National Park. That allows us to infer which sites may be best to sample which species. This study investigated the density in 3 muntjac species as they were able to observe how sexual maturity can change their morphology. And this is their density data in the unmarked animals using the random encounter model, which provides important information on the population. However, there are some limitations to traditional capture-recapture methods, so there is a shift towards using spatial capture-recapture, as it is able to incorporate the spatial organization of the sampling device and the spatial information in terms of the individual encounter data. And this is where Trap Tagger can help, as it enables the individual identification that can facilitate spatial capture-recapture studies. So these are some examples of those studies. This one found jaguar mating in terms of the seasonal changes on trails and their changes in the population size. So this is a camera trap image displaying the courtship behavior between the jaguars. And this is the density estimate across different days of sampling. This is a study that shows the evidence of mating for the leopards that the researchers were interested in, including this density curve of how it changes across the day. So these are important data that allows researchers to monitor the population dynamics, and for neuroscience purposes, it could be helpful for understanding whether there would be sufficient animals for studying their brain activity. So these studies that I have mentioned are a very good reference for me to understand how I could process camera trap datasets to get insights for designing potential neuroscience studies. As I mentioned before, for the snapshot Kruger dataset, I found the image with impala interactions that may be relevant to mating. So I also try to do some preliminary analysis on this dataset. This graph is showing the activity of how it changes across the day that could provide maybe the best period to get the most useful brain activity for my purposes into what period of time would be best to capture the mating interactions or other interactions that are relevant to the mating system. This is a community analysis showing the impala as well as other species present in the datasets that could provide a context of possible interspecies interactions that may be important for interpreting the brain activity results that researchers would get. There are also many other important information to know for neuroscientists to carry out their studies in the wild, including whether the species could habituate to the neural device, some possible sources of noise in the data that is collected, whether the species could undergo surgery and recovery, their diet, as well as the feasibility to collect other physiological data on this animal species. So from there are many next steps for my PhD. The first is to improve the quadruped model to incorporate more camera trap data images that would enable accurate post-estimation for training sexual behavior models, as well as understanding the sexual behavior more for the selected species to conduct neuroscience studies on. For the selected species, these will also need to go into a detailed camera trap analysis to further inform the design of the neuroscience studies. And the importance to incorporate an ethical and welfare framework, including the disturbances and the invasiveness to the species, that these framework could be observed through using behavior and neural measures as well. So there are many important progress that has been made in the neuroscience of sexual diversity, but these are limited to model organisms in the lab. And I'm giving some examples here. This one is done on the C. elegans in terms of the sexual behavior between the hermaphrodite and the male. And they did the brain-wide recording using calcium imaging. This study found how mating experiences changes synaptic connectivity in Drosophila females that was achieved through using genetic labeling and electron microscopy connectome. This one found a specific neuron subtype that can control mounting in same-sex sexual behavior, as they demonstrated this using optogenetics. So in light of these studies and the scientific advancements that has been made substantially in gene editing, in imaging, bioinformatics, high-throughput assays, automations, these same methods shown here could soon be deployed in wildlife animals as well. To finish, I would like to thank all of the participants and tutors, as I learned tremendously, which helped me advance this framework I'm using for my PhD. It is also the very first time that I spent so much of my waking hours with all of these participants and tutors during the meals, the outings, and working all together. And this was especially interesting in light of the load shedding situation in South Africa, where you get approximately 6 hours of power outage each day. So we'd had to withstand the very loud generators during these load shedding sessions. I'm also thankful to my partner for accompanying me to South Africa in light of the fallout of apartheid, which engendered South Africa to be currently ranked as the fourth highest crime rate of nations across the globe. And this was kind of observed when we visited Soweto, which is where District 9 was filmed, that discusses a little bit about these sort of issues. But overall, I really enjoyed my experience in South Africa with acquainting with some of its really rich ancient history, including the Table Mountains, which is the world's oldest mountain at 260 million years old. Then there's also the 2 billion years old Ridderfort crater that is large enough to see from outer space. And we also visited the Cradle of Humankind, which was including the 4 million year old hominin from the Sturfontein caves.